these different parasite strains and species are found in close proximity to one another. Now, let's consider the natural vector of Papatazi. Where is it distributed? So we're just overlaying where Phlebotomus papatazi, which is the vector of Leishmania major in black, is in fact found sympatric. We're found in the same biotopes as where Leishmania infantum are transmitted. Or let's go to India. In fact, in the same villages, in the same households where Phlebotomus argentipes is found and is the natural vector of Leishmania donovani in India, in that same household you can find Phlebotomus papatazi sandflies. But Phlebotomus papatazi has never been found infected with Leishmania donovani, so is not involved in transmission of Leishmania donovani. It's only involved in transmission of L major. Now, here's L major transmission in this part of Africa. It's a close related species of Phlebotomus fly called Phlebotomus dubowski, but it's not Phlebotomus papatazi. So there's a slightly different relationship between the sandflies found here and transmission of L major. Okay. So we'll spend some time trying to understand why sandflies are natural vectors of Leishmania and why we observe these interesting species-restricted vector competencies. In the laboratory, I want to make the point that this restriction of L major transmission by Phlebotomus papatazi in these regions, we can reproduce that restriction in the laboratory. So these are laboratory colonized flies, and we infect them artificially through a membrane. So the adult flies are in this cup here, and we put a blood meal in this vial. This is just mouse blood, and we seed that mouse blood with different parasites of different strains. So these are strains of L major and Leishmania mexicana, Leishmania tropica, Leishmania donovani. So lots of different strains of Leishmania, and the flies are artificially infected with these different strains or species of Leishmania. And if you dissect the flies, each of these points is the number of parasites in the midguts of the flies after just two days. And you can see that if you look early on, when the blood meal is still present in the midguts of these flies, that the parasites are present. It doesn't make any difference what strain you infect them with. But if you look later on in the midguts of these flies, when the blood meal has been digested by the fly and excreted from the gut, now only the L major strains persist. The other species have been lost along with the excreted blood meal. So you can reproduce that same sort of species-restricted vector competence in a laboratory setting. And now we're going to look at that in more detail and try and understand the nature of the parasite and sandfly molecules that mediate this specificity. So this is just a schematic of the life cycle of Leishmania major in its natural vector, Phlebotomus papatazi. So we're going to look at successive days after the fly takes an infected blood meal. So only female flies take blood. The blood is required for egg development in the female sandflies. Once the fly takes in a blood meal, whether it's infected with Leishmania or not, that blood meal is encased by a peritrophic membrane, which is a chitinous membrane, which separates the digesting blood meal from the the epithelial cells lining the gut, because there's lots of active digestive processes these are digestive enzymes, proteases and glycosidases that are released by midgut epithelial cells required for blood meal digestion. And this peritrophic membrane is thought to separate this digestive process from the epithelial cells lining the gut so as not to damage those cells during the digestive process. So this is where these parasites are, exposed to all these digestive enzymes in the posterior midgut. And so that's an important point. Once the infected blood meal is taken in, that blood meal goes to the posterior midgut, okay, and encased in this peritrophic membrane. Then the parasite and the fly, they produce a chitinase, which begins to hydrolyze, digest this peritrophic membrane, this chitinous membrane. So they produce a chitinase. Which allows at least some of the parasites to escape the confines of this peritrophic membrane. And they can move. Remember, they're flagellated, so they are propelled and access to the midgut wall, where some of them attach to the midgut wall. And we'll look at that attachment in more detail. And that occurs around day 3 or 4 days. And then these promastigates that are attached, the blood meal is excreted, the promastigates that have anchored themselves to the gut wall, they detach, and then they'll begin to migrate anteriorly and colonize the most anterior parts of the digestive tract. This is the foregut here. And this is the anterior midgut or thoracic midgut. And there's a valve here called a stomadeal valve, which is critical for the pumping action of the fly when it takes in blood and when it takes in sugars to get the contents of those feeds into the midgut. So there's this valve that separates the anterior midgut from the foregut. So it's interesting that there's heavy colonization that we'll observe more closely of the anterior midgut. There's not much colonization of the foregut because of this valve, but there's a huge accumulation of parasites behind this valve that separates the anterior regions of the midgut from the foregut. And then after a week or 10 days, if this fly is allowed to take another blood meal, the parasites that are in this anterior region of the gut will be transmitted, which is interesting because, again, these parasites have not colonized the saliva, so they're not injected into the skin when the fly salivates. They are propelled forward despite the fact that the fly is taking food, the blood meal, in this way. The parasites are propelled forward this way, and we'll discuss how that might happen. So all of these events are accompanied by the differentiation of these flagellated promasticate forms from these dividing procyclics to this elongated nectomonad form and to these mature forms that are found in the anterior gut, these haptomonad forms, and most importantly, these metacyclic promastigates, which are very motile forms, have this elongated flagellum in proportion to the cell body. So they're highly motile, highly active. And most importantly, these are the infectious forms. These are the forms that are deposited in the skin when the fly takes another blood meal. Okay, so I'll just show you some photographs of these events. So you can imagine these are blood-engorged sandflies feeding on a lesion on a mouse ear. So say this, and remember, Leishmania major is in fact a normal pathogen of rodents in the Middle East, different types of desert rodents. So you can imagine these are sandflies, Phlebotomus papatazi, feeding on a lesion on a rodent ear, and it takes in the blood meal into the posterior gut along with parasites. So here's a cross section of a blood meal that has been taken into the posterior midgut. And here is a nest of dividing promastigates. So they've differentiated from tissue amastigates that are in the macrophage. They've burst out of these macrophages, and now they're dividing as extracellular parasites. That occurs probably because of the lower temperature, the lower ambient temperature in which the flies are maintained at. These promastigates do best at 26 degrees, the infected cells themselves are probably destroyed during the proteolysis of all of this blood. So the amastigates are released. They rapidly transform to these dividing promastigates. If we do a cross section of the whole fly, we're looking here at the posterior midgut. So this is day 3 or something, and the blood meal contents have now been digested and excreted out. We can't see the hindgut, but the digested blood meal will be excreted. The parasites that are persisting in this gut after excretion of the blood meal, this is a higher magnification of persisting promastigates in the posterior midgut after the blood meal has been lost. And you could see that there's this very ordered attachment of promastigates to midgut epithelial cells at this time point. So those promastigates that can access the gut wall, they adhere very tightly via their flagellum to the epithelial cells lining the gut. So this is a video of these promastigates that are huge numbers that are attached to the midgut epithelial cells in this region of the gut after the blood meal has been excreted. So we're at this, and here's a Electron micrograph of a single promastigate, again with some association of its flagellum with the microvilli lining the midgut. And you could see how much larger this flagellum is relative to the size of the microvilli lining the gut. But still, it's thought that, so remember the flagellum is an anterior organelle. So it's pulling the parasite forward. It's not a tail. It's an anterior organelle. So the first association of the parasite with the gut wall will be via its flagellum, which is propelling it forward. The flagellum will insert between the microvilli of these epithelial cells. Okay. So now we're at a later time, probably at 7 days or 10 days post-infection. Again, the blood meal is gone. The parasites that were attached in this part of the gut seem to have been released, and now they've migrated to the anterior regions of the midgut. And huge numbers, just massive numbers of parasites colonize this anterior part of the thoracic midgut. Here's the valve, which has been compromised. It seems open here, and its function is compromised by this massive colonization of parasites in the anterior midgut. although there's still huge numbers of parasites even in this part of the midgut as well. So we're in this part of the midgut. Again, we're at least 1 week after infection. These are parasites that are stably labeled with RFP, red fluorescent protein. Yeah, it's a bit bright here, but you can see massive colonization of these red promasticates in the most anterior part of the midgut. This would be where the head of the fly was. It has been removed, and as a consequence, some of these promasticates that have colonized the anterior midgut, they're spilling out through this region where the head and the foregut would have been attached. There are still some promasticates left in the posterior regions of the gut, but there has clearly been this massive migration, anterior migration, of huge numbers of parasites to the most anterior regions of the gut. And again, this head has been pulled off. So now some of these promasticates, they're spilling out. They're swimming out of this region of the gut. Okay. So really, an impressive colonization of these flies. And it's that kind of maturation of infection with that kind of intensity of infection in the anterior gut, which is required for transmission by bite. So I want to emphasize again that all of these events are accompanied by these, the differentiation of these flagellated promasticate forms from these dividing procyclics that are found in the posterior midgut, and these are the first promasticate forms after amasticates transform into the extracellular flagellated form. There is this really interesting transformation at around day 3 or 4 to these nectomonads, which we and really nobody else is able to observe in culture. So this is such a beautiful promasticate flagellated form that we're really only able to appreciate if we look in vivo in the sandfly itself. These are poorly observed in culture forms. And then differentiation to these haptomonad forms, which are in the anterior gut. It's not clear what these do. These nectomonad forms are the ones that we think are critical for the attachment to the gut wall. And then the differentiation to the infective stage, these metacyclics, which have this impressive elongated flagellum, a very short and narrow cell body, and these are highly motile cells. These are the forms that are transmitted into the skin and initiate infection in the mammalian host. We've done some transcriptomic analysis of the in vivo promasticate stages. So I want to remind you that people who work on Leishmania, we're fortunate in that the insect stages are easily grown in cell-free cultures, in azenic cultures. We don't get to appreciate in vitro cultured forms all of the subtleties, the nuances of promasticate differentiation. But we do at least generate impressive propagation. They multiply beautifully, and you do get, at least in culture, some of them do differentiate to metacyclics. But in the sandflies, the differentiation to metacyclics is really impressive compared to culture. This is just an analysis because we are able to obtain in vivo promasticate forms, a transcriptomic analysis of the different forms, metacyclics, procyclics, nectomonads. This is just looking at these transcriptomic signatures, a principal component analysis. I just want to make the point that metacyclics, what this principal component 1 analysis tells us, that metacyclics are actually, their transcriptomes are actually closer to amastigates than are nectomonads or dividing, propagating procyclic forms. And what we now appreciate is that metacyclics are already beautifully pre-adapted to life in the mammalian host, that these changes in their transcriptomic signatures really occur prior to their being deposited in the skin, and they're already beautifully adapted for that environment. Okay, so going back to what goes on in these midguts, what are the potential barriers to the development of transmissible infections? So I mentioned this peritrophic matrix, this membrane that encases the blood meal, can act as a physical barrier to the migration of developing promastigates out of the midgut. So we now know that parasites make a chitinase to digest this membrane. The blood meal itself is flooded by digestive enzymes. So digestive enzymes induced by blood feeding, proteases and glycosidases that can inhibit the early survival, growth, and differentiation of promastigates. And the excretion of the blood meal remnants that can result in the loss of infection from the fly. So when the blood meal is gone, if the parasites have not somehow found a way to anchor themselves to the gut wall, they're going to be lost along with these blood meal remnants. Okay. So if we now consider these vectors which are very restricted in their vector competency, species restricted. So Papatazi, Phlebotomus papatazi will only transmit Leishmania major. What happens if you put another species in there? So I already showed you some of our experimental data. The loss of inappropriate Leishmania strains from non-permissive vectors occurs at the time of blood meal excretion. So the early colonization of the gut seems okay when you put different strains or species in these flies. As long as the blood meal is still there, the parasites seem to be persisting and growing. But at the time the blood meal remnants are excreted, that's when these inappropriate strains are lost, suggesting that attachment to the midgut is a critical event in species-restricted vector competence. So we and others have spent a lot of effort trying to understand the nature of the parasite molecules that mediate this attachment to the sandfly gut wall, and the nature of the parasite recognition sites that are expressed by the fly that mediate this attachment. So this is a schematic of the major Leishmania glycoconjugates. And what you'll note, similar to a lot of the majority of trypanosomatids and even pathogens like Leishmania, the majority of surface molecules are anchored in the membrane by these GPI anchors, these glycophosphoinositol anchored either proteins, GPI-anchored proteins, or in the case of Leishmania, glycolipids. And the major one is called lipophosphatoglycan. So again, it's a similar GPI anchor, but it's anchoring not a protein, but a glycolipid. And we'll look at this structure in more detail. And then there are also some related molecules, phosphoglycan-containing molecules that are released from or secreted by Leishmania. One is proteophosphoglycan, and there are some secreted proteins. So now I want you to take note of the commonalities of these surface-anchored and released the family of molecules that bear identical repeat units, these phosphoglycan repeat units, these yellow circles represented here. These are identical phosphoglycan structures that are either on a glycolipid molecule or are released in components of these proteophosphoglycan molecules, or even substitute bios on secreted proteins like acid phosphatase and acid phosphatase. Okay. So this repeat structure that are found in these different surface anchored and release molecules, the basic structure is a phosphorylated disaccharide containing a galactose and a beta-1,4 linkage with mannose. And these phosphorylated disaccharide structures are in a phosphodiester linkage, so you get a polymer of these phosphodisaccharide repeat units. And you can polymerize an impressive length of these repeat unit structures, either side chains here or as the major repeat unit structures of these LPG molecules. So if we look at the surface of Leishmania promasticates, it's this LPG that is the dominant structure, this structure here. There are between 6 and 10 million LPG structures per cell, and it forms this really impressive coat, surface glycocalyx structure that surrounds the entire cell, including the flagellum. So the entire cell, including the flagellum, is really, there is this tightly organized glycocalyx structure that is made up of millions of these GPI-anchored glycolipid structures. Okay. So what is the function of this LPG molecule? So again, this is the main structure. It has this GPI anchor. The LPG also has a glycan core structure that includes this galactofuranose, and then the phosphodisaccharide repeat unit structures that are polymerized by these phosphodiester linkages, and then a non-phosphorylated cap, which is a dimannoside non-phosphorylated cap structure. So these are the different structural domains of LPG. And a whole series of beautiful LPG biosynthetic mutants. So this is a remarkable oligosaccharide structure, which requires, as you can imagine, an impressive number of glycosyl transferases to generate this oligosaccharide. And in Steve Beverly's lab at Washington University and Sam Turco's lab at University of Kentucky, they generated and biochemically characterized a number of mutants of Leishmanias that are knocked out for, in this case, LPG1 was knocked out for the galactofuranosyl transferase that is required to put this galactofuranose structure on the glycan core unit. And they've knocked out a gene called LPG2, which is a transporter for the GDP mannose, the precursor sugar that is required. So required to get the precursor sugar into the Golgi to build this phosphodisaccharide repeat unit structure. So if we just consider the LPG1 mutant, it's because it's targeting just this structure which is on the glycan core, which is confined to LPG and not the other phosphoglycan molecules. This LPG1 mutant is deficient only in expression of LPG, so the major surface molecule is gone. The LPG2 mutant lacks the ability to polymerize, to generate these repeat unit structures on any of these surface and released phosphoglycan molecules. So putting these through flies, if we look at the LPG2 mutant, these mutants, they grow well enough so these are artificially membrane-fed sandflies seeded with these different promasticates or from these different mutants. The LPG2 mutant will grow well enough until around 2 to 3 days, at which point they're all killed, or the majority of them are killed. So the blood meal is still present in these flies at this time. So if we look at the LPG1 mutant, which is deficient just in the LPG1 surface structure, it grows until the time of blood meal excretion, and then those infections are lost. So this was using these mutants, and then these are the add-backs. These are the complemented parasites in which the LPG1 gene has been complemented and reconstituted. So this was the clearest indication that this dominant surface structure is required for these parasites to persist in infection at the time of blood meal excretion. And you can take the LPG that you purify from these parasites, and you can beautifully stain the microvilli lining the gut. So these are electron micrographic images of sections of the midgut and staining, beautiful staining of the LPG on the whole length of the microvilli structures. So LPG is clearly the critical molecule mediating the attachment, the adhesion of Leishmania promasticates to the gut wall. Absolutely required for the persistence of parasites in the gut and their full development. And then what we learned from an enormous amount of biochemistry, glycobiology done by many different groups, is that the LPGs from different Leishmania species vary in expression of these oligosaccharides. So turns out LPG is the most polymorphic Leishmania molecule that's been described. And the polymorphism, we don't think, is driven by anything that goes on in the mammalian host. It's driven by the diversity of receptors, parasite recognition molecules, on different species of sandflies. So we'll look at that in a bit more detail. But just for now, consider these remarkable polymorphisms in the nature of the LPG structure on different species of Leishmania. So they all have the same GPI anchor structure, the same oligosaccharide core structure. What they vary in are the nature and number of the repeats, these phosphorylated disaccharide repeats. They all have the basic phosphodisaccharide repeat unit structure, but some of them put these side chain sugars on these repeat units. So Leishmania major is polygalactosylated. The majority of those phosphodisaccharide repeats have side chain galactose structures. Leishmania tropica has a very complex branching oligosaccharide structure with glucose and arabinose as side chain structures. It's interesting that Donovani from Sudan has no side chain structures, has just this naked backbone. So still it has the basic LPG structure, but interestingly, no side chains. In India, most of the side chains are not substituted. Most of the repeat unit structures are not substituted, but a few of them do have glucose side chains. And then another polymorphism down here. And the New World strains look even different. So what drives this remarkable polymorphism in the structure of the major surface glycocalyx structure of Leishmania promasticates? Well, we think it's the nature of of the receptors, the parasite recognition sites that different species of flies express. So this summarizes, I know this is a lot of data. Let's just consider the first instance that we've been specifically talking about, this specific relationship between Phlebotomus papetazi and Leishmania major. So Phlebotomus papetazi, as I told you, will only transmit L major, and it's only L major that expresses these galactocele side unit structures, okay? Phlebotomus sargenti will only transmit L tropica. And again, the L tropica LPG on these parasites is again a highly branched oligosaccharide structure, but the side chain sugars are different. Again, they're mainly arabinose and glucose, not galactose. Now, when we come to flies like from India, Phlebotomus argentipes, which is the natural vector of Leishmania donomani in India, if we look at donomani in India, or we look at Leucemia longipalpus, the natural vector of L infantum transmission in Brazil, the parasites that these vectors transmit have a relatively simple unbranched LPG structure. Now, if we go back here, I'll remind you that these flies that naturally transmit these parasites with highly branched complex LPG structures, these are the restricted vectors. They will only transmit in the laboratory the parasites which they are the natural vectors of in nature. So this is where you see species-restricted vectorial capacity. The flies that transmit these parasites with simple unbranched LPG structure, these flies in the laboratory anyhow will transmit anything. They are broadly permissive. So this suggests that these LPG, these branched LPG structures, they put on these sugars in order to exploit sandflies which were available but had parasite recognition sites that could not accommodate simple unbranched LPG structures. So there was evolutionary pressure to express these sugar structures, in the case of L major, these polygalactosylated epitopes that could now bind to whatever the parasite recognition site was available to it on this fly, okay? The same with Phlebotomus sargenti. It had a parasite recognition site, some receptor, which required these structures in order for these parasites to colonize this fly. So these parasite recognition sites in the midgut of these flies, they're not there to bind these parasites. They're there probably as nutrient receptors for plant sugars and whatnot. And there's a diversity in these receptors depending upon the species of sandfly. And so that's what has driven this remarkable polymorphism in these oligosaccharide structures on this main surface molecule of LPG. Okay, so we're going back to now we're just going to look at the nature of the parasite recognition site on this Phlebotomus papetazi. So just to remind you that Phlebotomus papetazi will only transmit Leishmania major in these regions in black. So we were looking for a receptor on the midgut of these flies that had some specificity for galactose, because Leishmania major is unique in putting these galactose side chains on its LPG. And in fact, it was very easy to find on the midguts of Phlebotomus papetazi. We were able to clone out a tandem repeat galactin with high homology to mammalian galactins. So galactins are galactose binding molecules, And the carbohydrate binding domains exclusively bind beta-galactosides such as lactose and galactose. So this structure, again, it was a tandem repeat galactan. This is a linker unit in this tandem repeat. And again, a high homology with mammalian tandem repeat galactans. And this galactan expressed in Phlebotomus papetazi was restricted to Phlebotomus papetazi and the other natural vector of L. major, Phlebotomus dubosci. It was not seen in other species of vector sandflies. And it was confined to, so this is a Western blot analysis of mouse antibody against the recombinant galactan. And it was expressed only in the midgut. This is some staining, some confocal images of staining in red the galactan, which is luminally exposed in the gut and exposed in sort of patchy distributions on midgut epithelial cells. So again, this is the molecule that we were convinced was responsible for mediating the attachment of Leishmania major to the gut wall. And I'm just going to show one result of a transmission blocking vaccine. Because the point of all this vector biology perhaps is ultimately to identify targets of transmission blocking immunity. So we generated antibodies against the Phlebotomus papetazi galactan, and we infected the flies in a blood meal that contained antibodies against the galactan. And we looked at the number of parasites in the midgut at various days after feeding. And if we look at the time when the blood was still present, whether we had fed the flies on control antibodies or antibodies containing the anti-galactan antibodies, the numbers of parasites were the same. It was only at late time points after infection, when the blood meal was excreted, that we saw a clear effect of the anti-PP galactan antibodies, that it prevented, we presumed, attachment to the gut wall, and many of the parasites were passed. So this was one of the first, and remains one of the few examples of a target for a transmission blocking vaccine. So theoretically, you could immunize in hosts the problem is this is a zoonosis, so you'd have to immunize rodents. But imagining you could do that, you could immunize them with the recombinant galactan. They would make antibodies, and when the fly picked up an infection, it would also pick up antibodies, which would prevent the establishment of a mature infection in those flies. So that's an example of a transmission blocking vaccine. Clearly not a practical vaccine. You can't go about immunizing these non-human rodent reservoirs. Okay, so another point about the LPG molecule and its relationship to the behavior of these parasites in the midgut. So it mediates this critical attachment to the gut wall, but at some point these parasites have to detach and become free to migrate to the anterior regions of the midgut to position themselves for transmission by bite. So how does that occur? Is there some alteration in the structure of LPG? And again, work that was done by a number of individuals in Scotland and in Kentucky, looking at developmental staged alterations in LPG structure during metacyclogenesis. So this is the LPG structure on the procyclic forms from culture, which are the forms that develop in the fly, differentiate to nectomonads, and attach to the gut wall. During metacyclogenesis, 2 alterations occur. The number of repeat units gets larger. They approximately double in size. So this LPG coat becomes even more impressive. And the other critical modification that occurs, these galactosyl side chains become capped with arabinose. So these terminal galactose residues no longer become exposed, and they are no longer available for binding to these parasite recognition sites, which we've identified at least one of them, this galectin-like molecule on midgut epithelial cells. So this beautiful stage-specific modification in these side chain oligosaccharides permits these parasites to detach from the gut wall and migrate to the anterior regions of the gut. So the last point I'll make about the promastigote development and stage differentiation in the midgut is how do they become egested from the mouth parts of the fly? We've seen this beautiful accumulation of huge numbers of parasites, mainly metacyclics, in the most anterior regions of the midgut, behind this stomadeal valve here. But the parasites have to be deposited by some forceful action of the gut, forcing parasites forward, despite the fact that the fly is taking a blood meal the other way. So blood is coming this way, but the parasites have to go this way. So thermodynamically, how does that occur? It's not, I have to tell you, it's not, there's no conclusive evidence what goes on, but the best idea is this block fly hypothesis, that this massive accumulation of parasites behind this valve disrupts the normal feeding behavior of the fly. And it blocks the normal passage of blood into the midgut. And so they essentially regurgitate. They try and dislodge, they try and forcefully dislodge or vomit contents of this plug, this biological plug, forward. And that's how it's thought that parasites, particularly large numbers of parasites, get deposited in the skin when this infected fly takes a blood meal. And what facilitates that process is the secretion of one of these phosphoglycan-containing molecules called PSG, because it's this mucus-like material. It's this proteophosphoglycan that again has these phosphodisaccharide repeat units. So it's a critical content of this plug, this blockage, this plug that blocks the normal feeding behavior. Okay, so that is what is thought to permit large numbers of parasites to be deposited into the skin when this infected fly takes a bite. So just to summarize this bit regarding the development of promastigotes in the midgut, the surface and released proteophosphoglycans confer resistance to midgut proteases induced by blood feeding. This surface LPG, this beautiful glycocalyx structure on the surface of the parasite, mediates midgut adhesion, and the inter and intraspecies polymorphisms in LPG terminal sugars control the species and strain-specific restrictions of vector competence. So again, LPG is the most polymorphic molecule described in Leishmania, and it's driven not by not by factors in the mammalian host, but we're convinced by It's the diversity of parasite recognition sites in different species of sandfly vectors. The LPG polymorphisms are likely driven by the diversity of these parasite recognition sites, including these gut-associated lectins that are differentially expressed both between and within fly species. The developmental stage-specific modifications of LPG control the ability of metacyclic promasticates to detach from the midgut and migrate to the anterior midgut to position themselves for transmission by bite. And then lastly, I mentioned this proteophosphoglycan structure is a constituent of this plug, this biological plug, which is probably required for the regurgitation of parasites into the skin when the fly takes another feed. Why don't we just stop for a bit, and I'll take questions if you have any questions about any aspects of this vector biology.
My question is like, I was wondering about that different species of sandfly which you have shown all over the world. So is there any study which correlates to the availability of, like, they are mostly feeding on plant sap, sandflies. So has there been any study which shows the availability of food source and the Leishmania LPG structure?
Right.
Is there any study? I was like, you showed glucose, arabinose on the surface. It varies from different species. So the availability of food source in the different region all over the world, has it got any correlation to the parasite LPG structure?
Yeah, that's an interesting point. The diversity of these parasite recognition sites, at least in the case of Phlebotomus papatazzi, is this lectin, this galectin-like molecule, which again we're fairly certain it's not there as an attachment site for Leishmania major. It has nothing to do with that. It's there for some other function. Lectins can serve a defensive purpose, or it may be a receptor for particular sugars for nutrient acquisition. So no studies have been done looking at different. So the problem is the only receptor that has so far been identified, receptor for Leishmania, is this work on Phlebotomus papatazzi identifying this galectin. So what are the receptors for parasites in the other flies? So we don't know what they are, but your point is, are they different because they're feeding on different sugars? So we're going to talk a bit more about the physiology of these sandflies in terms of sugar acquisition in the discussion now, but it's an important point. After these female flies take a blood meal, they're then maintained on sugars. So both male and female sandflies, their carbon sources are various plant sugars, either saps or nectars. And so depending upon their biotopes, the nature of those plant sugars will be different. And that's what we presume leads to these different potential recognition sites for these parasite LPG oligosaccharide structures. But again, none have been identified except for with this work in Phlebotomus papatazzi. And why Phlebotomus papatazzi should have this galectin and not other flies, I have no idea. And it's distributed across such a wide biotope, from North Africa all the way to India. And we've looked at a few flies from these different geographic sites, and they all have the galectin. So it's interesting to consider, what does the galectin do in Phlebotomus papetazi, and why is it not required in other sandflies? But it was clearly exploited by L major. It serves as a beautiful recognition site so long as the parasite could put on these polygalactosylated LPG structures.
Thank you.
How crucial it is, the length of flagella of this parasite in causing the infection, because we saw that the length of the flagella varies in different stages. So during these stages, if the length of flagella during the attachment, is there any crucial role of flagella in the attachment of to the midgut epithelial wall, or causing to the infection? Because if there is any, or what is the mechanism behind this varying length of flagella which is occurring?
Yeah, well, that's an interesting question, too. I don't have an answer for that. No one's looked at that. It's this interesting relationship. Is the elongation of the flagellum at the cost of the size of the cell body? So when the flagellum is very long, the cell body is in fact very foreshortened. So in metacyclics, the cell body is really very small. Yeah, that's an interesting question. If we look at these parasites again. Yeah, well, nectomonads have an impressive, but in this case, the flagellum serves a very useful purpose. It propels it forward and is where the attachment is. So we were initially convinced that the critical molecules mediating the attachment would be confined to the flagellum, because that's the nature of the attachment that you see. But I think it's only because it's an anterior organelle, and the LPG, in fact, encompasses the entire cell, including the flagellum. So and it's only the flagellum that is small enough to insert itself between microvilli. But you know, these old images from the 60s and 70s showed that this attachment to the gut wall was always via the flagellum. So everyone always assumed that the critical molecule was confined to the flagellum. So I'm quite convinced that's not true. It's simply a thermodynamic thing. It's an anterior organelle. That's what encounters the gut wall first. So that's where the attachment is. But the length of the flagellum, your question is, yeah, I mean, it's, this is such an impressive, I think the motility of metacyclics is so impressive. It probably is critical in their ability to be transmitted by bite and to migrate to the anterior parts of the midgut. So these are the forms that really have to migrate. They have to move forward. One question is, why do they move forward? And we'll look at, they move forward because that's where the sugar meal is. That's where when the blood meal is gone, that's where the carbon source is in highest concentration. Because the sugar meals are stored in the crop, which is diffused into the anterior midgut. So we think they're following a sugar concentration. That's why they move forward. But it's the propelling action of the flagellum which allows them to move.
So you're saying that there is a chemotactic movement towards the sugar gradient, that is taking place?
Yeah, so we'll look at that a bit more now. Okay. But you know, structurally, it's so interesting, your question. how these structural changes in the cell body and in the length of the flagellum, how that is regulated, how that occurs. So no one has studied that. And really, unless you have sandflies, you really, most people don't appreciate this phenomenal heterogeneity in the morphology of promasticate forms. If you look carefully in culture, you can at least see some of these, but this other heterogeneity is not so clear. But these are very precise developmental stages that occur in the fly, and that are not so easy to appreciate in culture.
In LPG knockout mice, in LPG knockout parasites,
Yeah.
how they activate host immune response?
Well, so first of all, if you look at that, the flies can't transmit them because the LPG knockouts will not be transmitted by flies, right? They're lost from those flies. But you can inject them by needle. And they don't survive. It's an interesting question. They do not survive very well in the host. Not so well. So the LPG2 knockouts, they really don't survive at all. The LPG1s, deficient just in LPG, they survive a bit, but they clearly have a problem. And we'll consider, I guess, tomorrow, some other roles that LPG plays in the mammalian host, in macrophages. Okay. Yeah.
Sir, what is the difference between this metacyclic and nectomolar? That the metacyclic is very infective, as in case with nectomolar. Sir, that I have a question.
Yeah, it's a very good question. Difference between here and here. So importantly, they are both non-dividing. They both stepped out of DNA synthetic cycle. They are resting cells. But this is not infective. This one, by infective, this will not do well inside of a macrophage or in a mouse. This one is just beautifully adapted, pre-adapted to the mammalian host. This one is not, but transcriptionally it looks more like procyclics, and metacyclics look quite a bit more like amasticates, in tissue amasticates. But there are some really interesting transcriptional differences between this form and this form. A lot of them have to do with cell cycle changes because, again, this is a resting form. Yeah.
Sir, 20 to 30 species, they are able to cause Leishmania, but rest of they are not. Sir, why so? Means, what is the difference between that species of that causes the leishmaniasis and that doesn't cause?
You mean there's so many different species of sandflies, most of them do not transmit. So I don't know the answer to that. I think there's like at least 120 different species of sandflies. Most of them have not been found infected. For a lot of them, it may be because they really are not found in the same biotope as the parasite, or their blood meal sources do not include hosts that are infected with Leishmania, or they have barriers to development. They may not have parasite recognition sites so the parasite may not be able to attach. It's similar, I suppose you can extend the question to why do mosquitoes not transmit Leishmania? So we have infected mosquitoes with Leishmania, and they grow okay as long as the blood meal is present. When the blood meal is gone, we never see infection persisting. So this attachment may be the key thing that prevents mosquitoes from transmitting Leishmania. Then just the lack of suitable recognition sites. But then you would have thought, well, why hasn't the parasite, just co-evolved some expression of molecules on its surface so it could take advantage of mosquitoes for maintaining a transmission cycle? Interesting question. So I'm sure there are a number of other barriers to transmission by mosquitoes and other sandfly species. But it's a nice question. We've never had a sandfly species to work with in the laboratory that is not able to transmit, to address that question. At what point during infection are the infections lost? Right. Okay, another question?
Sir, actually, if...
They really want you to use the microphone.
Sir, actually, this is where, I don't know, this is a very simple question. If we introduce the Northeast Indian species of Leishmaniasis in the Maharashtra state, does it cause Leishmaniasis, or it survive in this place or this environment? Or if the Leishmania, this sandfly of Maharashtra state, if we introduce this sandfly to the Northeast area, which does not carry the Leishmaniasis?
Well, no, I think so. It's hard to remember all these slides I showed you. But so Phlebotomus papetazi that transmit L major is in, I know it transmits L major in Rajasthan. So, but Phlebotomus papetazi is also found in the Kala-Azar endemic zones. And I told you, in the same household that they go in and get Phlebotomus argentipes and found infected with Leishmania donovani, that same household, you'll find Phlebotomus papetazi. So they've had an opportunity to pick up, they clearly have had an opportunity to pick up Leishmania donovani, but it just doesn't work. It doesn't work. In fact, it's the India experience that is most forceful in demonstrating in a natural setting that Phlebotomus papetazi really will not transmit anything except Leishmania major, because you find them in the same endemic household where you find an active case of Kala-Azar. And you find human blood in them, so their blood source, can be other animals, but they like to feed on people, so it just doesn't work. And in the laboratory, we can show that it just doesn't work. Okay.
Sir, one general question: why Leishmania major is called major? Why Leishmania major is called major? Maybe because it affects in many countries or some any other region.
Oh, why is it called Leishmania major? I have no idea. You tell me. Make up a reason. I'm sure that can be your project. Why was it called? No, historically, I'm sure we could go back and find. I have no idea what. All right, let's finish up with a discussion of the microbiome in these sandflies. Everyone's interested in microbiome. And clearly, these insects, mosquitoes, have a very interesting microbiota, natural gut flora. Because, look, we're talking about Leishmania parasites that are developing in the digestive tract. So clearly, there's some microbiota that they're cohabiting this environment with. So does the natural midgut microbiota inhibit or promote Leishmania development? So in sandflies, there's not a lot of work done on the natural gut flora, but We know that it's acquired by adult sandflies from several sources, including during feeding on their respective animal and plant sources of blood and sugar, or from recolonization of the gut by the microbes ingested by the terrestrial dwelling larval stages. Sandflies, unlike mosquitoes, their larval stages are terrestrial. They're feeding on organic material, like feces, and in fact, we maintain our sandfly cycles, our larval stages of sandflies, on aged rabbit droppings and rabbit food. That's what the sandfly larvae like to eat. So clearly, the midguts of larvae and some of that microbiota will be retained in the adult stages. So there's an interesting microflora in sandflies. A lot of work has been done on the natural midgut flora in other pathogen vectors. So tsetse flies that transmit trypanosomes, if they get rid of the natural midgut flora, the establishment of the infection is promoted. That's same with mosquitoes infected with malaria, mosquitoes infected with dengue virus. In each case, if you get rid of the midgut flora, the infection by the pathogen is improved. You get a more forceful colonization of those mosquitoes or tsetse flies. But something quite opposite happened when, so this was an obvious experiment to try in sandflies and Leishmania. Let's just treat the flies with antibiotic. We just treated them, we included penicillin and streptomycin in the blood meal and or sugar meal, because again, once the blood meal is gone, you have to maintain the flies on a source of sugar, and we just give them 30% sucrose, and that's what the flies are maintained on. But when we included antibiotics in the blood and the sugar meal, we found quite the opposite of what we expected. So we're just looking at the mature metacyclic forms, which are only found late in infection, day 9 or day 14. But in the pen strep, the antibiotic-treated flies, no metacyclics. I mean, there were none there. And this just shows that if we included the antibiotic in the sugar and the blood meal, it prevented development of metacyclics. If it was only in the sugar meal, after the blood meal is gone, that also prevented metacyclogenesis. If we just put it in the blood meal and then excluded antibiotic from sugar meal, the metacyclics were able to develop. So antibiotic, which is producing dysbiosis of the normal gut flora in these flies, had this profound effect on the ability of Leishmania infections to develop normally and mature and generate the infectious stage metacyclics. So we did a lot of work trying to figure out, well, why does Leishmania need these bacteria in the gut? Well, first of all, we just did some, we characterized the gut bacterial size and diversity by 16S sequencing, just by culture on agar. The antibiotic-treated flies, their midguts had, log-fold less numbers of bacterial colonies. That's not surprising. By looking at the diversity, as you can imagine, there were bacteria in the antibiotic-treated flies that replaced the normal midgut microbiota. The lists are shown here. Again, their numbers were a lot less, and their diversity had changed. These were clearly they were displaced by bacteria populations that were resistant to the antibiotic. Well, what we did find was that if we took our antibiotic-treated flies that did not permit the development of the proper numbers of metacyclics in the anterior gut, and we fed those antibiotic-treated flies on bacteria that on some bacterial populations from the normal gut flora that we made resistant to the antibiotic, either Leishmania, Rhinella species, Enterobacter, Serratia. In each case, they could reconstitute at least a little bit the ability of the antibiotic-treated flies to support the development of metacyclics. So it didn't seem to make so much difference what bacteria, as long as you reconstituted at least some of the normal bacterial populations in the gut. The parasite would develop better. So I'm going to tell you now, we still don't really know why the bacteria are so essential to Leishmania development, but I'll just present what we think is going on. This comes to the issue of sugar meals. So our flies are maintained on 30% sucrose. And our flies, and most people who have sandfly colonies, we maintain our laboratory flies on 30% sucrose, which more or less matches the concentration of sugars found in the natural nectars and saps that sandflies normally feed on when they feed on plant sugars, okay? That's the concentration of the various sugars in normal plant sugars. So that's why we use 30% sucrose. So when a blood meal when female flies take a blood meal, the blood meal is shunted to the posterior midgut. But when flies feed on sugars, the sugar meals are in fact not shunted to the posterior midgut. They're stored in this bag called a crop, which passively diffuses the sugar contents into this is the anterior midgut. This is where the metacyclics accumulate, right here. So the sugar passively diffuses there. The fly utilizes the sugars, the bacteria utilize the sugars, and the parasite utilizes the sugars as a critical carbon source for energy. But the sugars are stored here. So this is a higher resolution image of this crop and this duct that allows passive diffusion of the sugar meal into this anterior midgut here. And this again is the critical area where the metacyclics accumulate in the sandfly. What we observed if we, I'm missing a slide. What we found is if we take metacyclics and we expose them to 30% sugar in vitro, they're killed within seconds by these hyperosmotic conditions. So that seems strange because in the fly itself, metacyclic station here, they're going to be exposed to these really high concentrations of sucrose that we've fed the flies on. And yet in vitro, you kill the promasticates instantaneously by these osmotic stress conditions. So we think that's what the bacteria are for. They permit the parasite that have accumulated in this region of the gut to tolerate these hyperosmotic conditions by competing for sugars. Their metabolic needs compete with the fly and the parasite for sugar consumption, diminishing the concentration of sugars to a tolerable osmotic level for the parasite. And we could sort of convince ourselves that was true if we maintain the flies on 15% sucrose instead of 30, which the flies hate. So most of the flies die because they need a higher concentration of sugars. But the ones that survived, the antibiotic-treated flies didn't kill the parasites so much. They permitted better development of metacyclics. And here's 10% sucrose. Even fewer, but again, so majority of flies died, but the ones that survived, at least we could find in antibiotic-treated flies, they supported metacyclic development better. So these are some time-lapse images of the plug region, this anterior midgut region. So huge numbers of, these are red parasites in the plug. And this is time-lapse imaging. You can see how active the metacyclics are. If these lights, actually, could we just turn these lights out? 'Cause, well, I could have done that. Yeah. See how beautifully active? So these are mainly metacyclics in this plug region. And they're not attached, they're swimming around. They're just sort of waiting. They're waiting for the fly to take a bite so that they can be egested into the skin. So now this is an antibiotic-treated fly. So we've dysbiosed the fly. We've killed off most of the microbiota. The parasites still form a plug, and there's still parasites here. But if we look at this portion of the, so this is a video image, but there's deformed parasites, and they're not moving so well. So we think they're dead. They've been killed by, we think they were exposure to this high concentration of sugars. If you look just at this region of the gut, not so far back, these parasites are perfectly happy, okay? So, I don't know, that's what we think is going on. Another, these are other images showing this same thing. Again, here, perfectly happy here, and this is the plug region. Really, these are deformed parasites, no movement at all. These are all dead. So they've gotten there. The metacyclics have gotten to this part of the gut, but they've been killed. We presume by this exposure to such high concentrations of sugar coming in from the crop. Now, we also presume the bacteria could be providing essential nutrients for parasite development, but we were unable to conclusively show that. So again, we have other, there may well be other contributions of the normal midgut microbiota to Leishmania development, but just to summarize, the bacterial communities naturally present in the fly midgut are essential to the survival of infected stage metacyclic promistigotes in the anterior gut. The role of the microbiota in L major development remains unclear, though evidence points to their competing demands for sucrose to promote osmotic conditions compatible with promistigate survival. But more generally, changes in the size and diversity of the bacterial communities will profoundly influence vector competence. So it'll be interesting for other people that have flies, either natural caught flies or colonized flies, to look more carefully at the nature of their bacterial communities, their normal gut microbiota. Okay, we can take some questions on this, and then we'll have a break. A few questions just on this part.
Sir, the Leishmania causes any harm to sandfly? If sandfly is infected with the Leishmaniasis, and if it get a blood which is not with the leishmaniasis, does it cause leishmania cause any changes in the body of sandfly? Means a type of a disease.
Right, so that's an interesting question. Clearly, heavily infected flies have trouble taking a second blood meal, because they would have already had one blood meal. That's how they got infected. And remember, female flies take blood in order for egg development. So after the first blood meal, they would have laid their eggs. The fact is, in a natural setting, after a single blood meal, when that female lays the eggs, a lot of them die. Clearly, some do survive to take a second blood meal. Otherwise, we would never have transmission of Leishmania. Transmission requires at least 2 blood meals, right, for infection and then transmission. People have looked at the reproductive fitness, the numbers of eggs and the viability of eggs laid in an infected and non-infected fly. At least after the primary infection, there doesn't seem to be much of a consequence. But clearly their ability to take a second blood meal is probably affected. But the frequency of sandflies that are infected with Leishmania in even hyperendemic settings is quite low. So these parasites don't contribute much to the reproductive fitness of the population of sandflies in these biotopes. That's not thought to be so. Yeah.
I have a question like whether the microbiome of the sandfly gut, while it regurgitates the parasite into the bloodstream, and macrophages are the host. So does the microbiome, like the way it regurgitates, the saliva must be containing microflora. So is it the reason why it is choosing macrophage as a host? Like,
Yeah, so that's a lovely question. So the question pertains to these parasites we think get transmitted because the fly, remember, the parasites are not in the saliva. They're still in the digestive tract. They're being regurgitated forward. So does that mean that other contents of the gut are also regurgitated like the normal microbiota? And that's almost certainly true, and now there's some recent data to suggest that, and we'll discuss that tomorrow, whatever day. We will talk about that. That's a really interesting point. So flies aren't just depositing parasites, they're depositing, well, the flies do salivate, so contents of saliva, and they will also be depositing bacteria and perhaps fungi into the skin. So that can profoundly affect the nature of the innate responses, which we're discussing tomorrow, if we're all alive tomorrow. We'll be here tomorrow, right?
So does the nutritional status of the host, would that determine, because nowadays we are having the diabetes on rise. So many people are, they have high sugars in their blood. So I mean, this is just a weird question, but.
Flies would like that, I think. So.
Sorry?
Flies would like that. So. No, it's probably an interesting fact variable to think about. Okay, we're taking a
short break.
Well, we go to 11 o'clock? So we'll be back at 11. Okay. I had a question during the break about a very pertinent question about immune response pathways in insects, and which are so beautifully described. I told you yesterday that a Nobel Prize was recently given because of the innate immune response pathways described in Drosophila by Jules Hoffman, from which these toll-like receptor pathways in mammalian cells were then described. So there's clearly some relevance in terms of these insect immune responses, which mainly evolved in the context of bacteria and fungi, not so much in terms of eukaryotic pathogens like Leishmania, but it's still a relevant question. Do Leishmania trigger an immune response pathway in insects? And if they do, does it influence Leishmania development? So again, there's two pathways in insects, the toll NF-kappa B pathway, which, again, the toll receptors in insects are also involved in dorsal ventral patterning. So if you knock out any of these components in the toll pathway, it's lethal. So it's difficult to study. But this pathway, again, it's similar to the toll-like receptor pathway in mammalian cells, leading to NF-kappa B translocation to the nucleus and activation of immune response genes. In insects, it's activation of antimicrobial peptides, these AMPs. The IMD pathway, the immune response pathway, the alternative immune response pathway in insects, is not devoted to any other function other than innate immunity. So you can knock out components of these pathways and it's not lethal. And it's involved in, again, the transcription factor here is relish, and it's involved again in the upregulation of genes coding for antimicrobial peptides that kill gram-negative bacteria. So I'll only mention that our own work and a couple of other labs have looked at antimicrobial peptides in immunity to Leishmania. They've looked at defensins, that's one antimicrobial. They're certainly upregulated in Leishmania-infected sandflies, and there seems to be some sensitivity to Promaster. We use CRISPR-Cas9 editing to knock out relish, this transcription factor in the IMD pathway. And the flies developed higher levels of Leishmania infection. I'm suggesting that the IMD pathway does promote some level of innate resistance in sandflies against Leishmania. Okay, so we're going to shift now to some parasite genetics. So we're not completely leaving sandflies behind because we're going to talk about genetic exchange or sex in Leishmania, and that takes place in the sandfly. And I did want to acknowledge a lot of these, all of this work was done, and a lot of it's not yet published, was done by Ehud Inbar, and the bioinformatics that I'll discuss is done by Jahangir Shaikh. Okay. Okay. Well, here's that slide again that emphasizes the diversity of clinical forms of disease and the diversity parasite species that produced these clinical outcomes, and the diversity of different hosts that can be infected. So a huge amount of diversity that is observed in the Leishmania genus, and all transmitted by these flies. So, so we're interested in how did all this diversity arise? All these different species and strains that produce such different clinical forms of disease and different mammalian hosts that can be infected by this parasite. How did all this diversity arrive? Then it was assumed that it arose very slowly over tens of thousands, millions of years by the accumulation of somatic mutations. And sex or sexual recombination was not thought to be so important if it occurred at all. So we're going to consider that question. Just some general points about the Leishmania genome. So again, this is a unicellular eukaryotic pathogen in the class Kinetoplastida. The nuclear genome, for most strains, it's 36 pairs of chromosomes. And those are all the strains from the old world, like Leishmania donovani. Interestingly, Leishmania mexicana, a New World strain that produces cutaneous Leishmania, it has only 34 chromosomes. Leishmania brasiliensis, that produces these mucosal lesions, is 35 chromosomes. These organisms are diploid, but with lots of aneuploidy. So they're largely diploid, but with all this interesting aneuploidy, and we'll talk about this aneuploidy. And these, again, they're in the class Kinetoplastida. That means that they have these kinetoplasts, which are essentially the mitochondria of these parasites. And the kinetoplast DNA is a network containing thousands of catenated DNA molecules. These circular DNA molecules, both maxi-circles and mini-circles, that represent 10 to 20% of the total DNA content. But we're really going to talk mainly about the nuclear genome. So this genome plasticity and aneuploidy that I mentioned is one of the more recent described features of the Leishmania genome that is so interesting. So this is a review paper by Sturkers, these are just various strains, Donovani strains and major strains and Mexican, different strains and species. They looked at that depth breeds of whole genome sequences of these parasite genomes to ascertain the chromosome copy number of the 35 or 34, 35 or 36 different chromosomes that are listed here, okay? And this is a heat map, so the different colors represent the SOMI levels for each of the different chromosomes. So blue is disomic. So there's a generally blue pattern because these parasites are generally diploid, but they're not, you know, they're clearly, there's a lot of aneuploidy going on here. Primarily, a lot of chromosomes are not disomic, but trisomic. These green colors represent trisomic chromosomes. Some of them monosomic, these dark colors here. And chromosome 31 has been well described for many years now as being tetrasomic. So its normal copy number is 4. Four copies of chromosome 31. So if you look at everything all together, they're more or less diploid with enormous amount of aneuploidy. So these parasites seem to tolerate a huge amount of aneuploidy. Now, most higher eukaryotes, they don't tolerate aneuploidy at all. Of course, in human populations, you know, aneuploid gives rise to some pretty serious congenital malformations, like trisomy 21 is Down syndrome. But these eukaryotic single-celled organisms tolerate this fantastic level of variation in chromosome copy number. So why do Leishmania tolerate aneuploidy? It can be considered as a non-Mendelian means to create genomic variation when recombination is absent or minimal. Variation in chromosome copy number may also regulate gene dosage as an alternative to normal transcriptional control. So Leishmania, as well as other trypanosomatids like African trypanosomes, and they don't have normal transcriptional control. There are no promoter regions that dictate when a gene should be turned on or off. There's constitutive transcription of these polysomic RNAs. And transcriptional levels are mainly controlled by the regulation of gene expression, again, does not take place at transcription, which is constitutive in these polysomes, but instead the Leishmania rely on post-transcriptional degradation of these messenger RNAs and also translational control. So just by changing the gene dosage, you can change the level of the relative level of mRNA abundance by simply doubling or halving the number of chromosomes that are being constitutively transcribed. So again, it's not so clear why these parasites have evolved and tolerated all of this aneuploidy, but that's one of the thoughts behind it. It's an alternative to normal transcriptional regulation. So we're going to see in the discussion now, we're going to see a lot more aneuploidy, and I just want you to keep that in mind. So as we discuss reproductive strategies, which are still sort of under debate for this parasite. And the debate, of course, involves clonality versus sexuality. If we look at the life cycle that we've described so far, it appears to be asexual. That we've described dividing forms in the midgut of sandflies and dividing forms in the intracellular endosomal compartments of infected cells. So these are asexual clonal propagation. And there's no obvious sexual dimorphism. If there is sex, it's not, there's not any obvious sexual stage that distinguishes a male gamete and a female gamete in either of these hosts. So everyone assumed for a long time these were just asexual parasites. But we come back to the central question, well, how did all this diversity arise? How did we generate so many different kinds of species and strains if it's just clonal? It can absolutely happen over a long, long time, just an accumulation of somatic mutations. And certainly the population genetics that was done using a limited number of markers, the Leishmania were thought to be primarily, if not exclusively, clonal. So this is a seminal piece of work by Thiberank and Ayala, the French group, that put forth this clonal theory of parasitic protozoa, that they were essentially clonal, and based on what they said was either no mating or only rare sexual mating in natural populations supported by high linkage disequilibrium. And by that means non-random association of genotypes occurring at different loci. Sexual populations that are intermixing normally have very low linkage disequilibrium. So looking at their analysis of marker loci convinced them that these were asexual, these were clonal based on high linkage disequilibrium. But as people were using more discriminating markers and collecting more Leishmania isolates, people were continually finding genomes that looked to them like they were hybrids, that they, consistent with intermixing, consistent with genetic exchange, because based on these markers, they could describe hybrid genotypes between different species. So remember, I told you there are at least 20 or 30 different Leishmania species. That's based on the collection of various unlinked markers across the nuclear genome. And then they described isolates that seemed to be a mixture of two different species. And then our own work, a few years back now, because we had sandflies, and we were convinced if sex happened, it probably involved the extracellular stages of the parasite. And that would occur in the sandfly host, the sandfly vector. And that's in fact what we found. We could formally demonstrate genetic exchange involving promasticate stages as they develop in the midgut of flies. Okay? So this is just a slide showing Leishmania hybrids in the real world. By hybrids, again, looking at markers distinguishing different Leishmania species, they found isolates that appeared to be a combination of two species. The parasites had, consistent with outcrossing, two species having had sex. So in South America, hybrids between Braziliensis and Panamensis, Braziliensis, Guianensis peruviana. In the New World, hybrids between L. major and Arabica. Let's see, and in India, Leishmania tropica, which is up here in India, not here. Two different strains of tropica. And let's see, in this region, Leishmania donomoni, hybridization between two different strains of L. tropica. So lots and lots of reports suggesting that these parasites have the machinery for genetic exchange. And our own work in sandflies formally demonstrated that they do. So this was the strategy for demonstrating the generation of experimental hybrids. What we did, we introduced independent drug resistance markers into two different lines of L. major. So this is an L. major strain from Israel. This is an L. major strain from southern Russia. So they're both L. majors, but they have a lot of different SNP polymorphisms, which we'll look at. And we introduced drug-resistant markers, two different drug resistance markers, one to the antibiotic SAT, into the SSU locus on chromosome 27 here, and the other strain, it was a replacement construct of an LPG by a synthetic gene in chromosome 24, so knocking out LPG, an LPG biacidic gene, and now there was an expression of a hygromycin resistance marker. So we took these two transformed parasite lines, we co-infected flies with both of them, and then you dissect out the midguts, and then you submit those promastigotes in the midgut to both antibiotics. And only the parasites that have had sex or had undergone genetic exchange and express both antibiotic resistance markers will grow. And that's what we were able to fairly easily demonstrate. Not in all the flies, but in a high proportion of flies, we could easily grow out double drug resistant parasites. So that is the strategy that we use to generate hybrids that we co-infect flies with two different strains that each having different antibiotic resistance marker and submit them to double drug selection. So I'm just going to summarize some years of work showing the different strains that we've been able to hybridize in the sandflies. We can take, these are L major strains from across the entire geographic range of Leishmania major. And in pairwise combinations, we could get them to have sex in these flies. And these are L. tropica strains. We could get different L. tropica strains to have sex with each other. And in red, this is interesting because we could cross-species hybridize. We can take two different species, L. major and L. infantum from Spain, and even L. major and amazonensis from Brazil, which are genetically quite distant, and they would still cross-hybridize. So that's interesting because they're different species. Usually the definition of a species is that their ability to have sex with is confined to members of the same species. So we may have to sort of rethink what we mean by species, since we can mix any species and they can still have sex. So mating was not restricted to specific strains or pairwise combinations. There's no evidence for mating types that might limit genetic exchange. So if you look at other lower eukaryotes, if you look at fungi, they also have a sexual cycle, which usually involves different mating types. You can call them a male and a female mating type. In fungi, it's called alpha, A and alpha mating types. That restrict the ability of fungi, of spores, to undergo meiosis. Well, we didn't, so far, we have no evidence that there are mating types. We can just throw different strains together, and at least some of them will have sex. So experimentally, there is no species barrier to mating between species. It supports the likelihood that observed natural hybrids are products of sexual outcrossing events. But based on the frequencies of co-infected flies yielding hybrids, sex must be considered non-obligatory and relatively rare. So actually, the majority of flies in our system, we can't recover any hybrids. And yet, the parasites seem to undergo full development. So it's not an obligatory part of the life cycle. That said, our flies may be very artificial. They've been in colony. We've had them colonized over hundreds and hundreds of generations over many, many years. So they may not support these genetic exchange events so efficiently as natural flies. So we just don't know. But clearly the parasites have retained the machinery to have sex, even though they seem not to do it so frequently. It raises the question, well, why in some flies does it happen and not in others? So we don't know the answer to that. Now, importantly, multiple attempts to generate hybrids in vitro or in the mouse were unsuccessful. So taking these same constructs, these drug-resistant constructs, mixing them together in culture, we couldn't get them to have sex. And injecting both strains into a mouse, either the skin or the footpad, or we couldn't select for double drug-resistant parasites. So the sandfly is where sex happens so far. So there is some condition in the fly. And I've already shown you that this beautiful development of promasticates through these different morphologic forms, procyclics, nectomonads, haptomonads, metacyclics. We see that in flies, and we don't see that well in culture at all. So already we know that what we grow in culture is not approximating so well the real conditions in the midgut of these flies. And this is another demonstration that that's true. We can get these parasites to have sex in flies, but not in culture. So when and where does mating occur in the flies? So these are the different forms of the parasite that we've talked about. And so now we're just dissecting out infected, the midguts, these flies that have been co-infected with these two drug-resistant lines, and that are then submitted to double drug selection pressure. If we dissect the midguts at day 1 or day 2, when we have really only procyclic forms, we've never recovered hybrids. But already by day 3 or 4, when we see these nectomonads, we can recover hybrids. And the longer we wait, the more hybrids we recover. So the sexually competent forms might begin here, but again, the longer, these all may be sexually competent. Okay, so I'm going to emphasize we still have yet to visualize cell fusion events. We still have yet to visualize these parasites actually having sex. So we haven't identified gametes or a haploid gamete stage, and we've yet to visualize these cell fusion events. So these are the sorts of things that we're keen to do now. So we know they've had sex, and now I'm going to go through the whole genomic sequencing to look at the kinds of inheritance patterns that occur when we select for these hybrid parasites using these drug-resistant lines. So the question is, do they have a true sexual cycle involving meiosis, gamete formation, and sexual recombination involving reciprocal crossover events during meiosis between homologous chromosomes? So we're going to address this by a genomic analysis, because again, we can't see these events. We have yet to identify an actual meiotic stage. A lot of the stuff we're seeing in Leishmania, it's very interesting to look at the fungal literature, because they, particularly in terms of aneuploid genomes, which are so prevalent in Leishmania, this aneuploidy, that has been well described in yeast, and it can arise by a sexual reproductive strategy that's been described in Cryptococcus neoformans, that they do have a true sexual cycle involving meiosis, but they see a fair amount of aneuploid. But what was most interesting to us is this parasexual cycle described in Candida albicans, in which these aneuploid products of genetic exchange are really well described. And parasexual process just involves fusion of two cells and fusion of the nuclear DNA. And then a random or concerted loss of chromosomes. So it doesn't involve meiosis. And just after these fusion events of diploid cells, the chromosomes are randomly lost, and you end up with a some reduction, which generates a lot of aneuploidy. And that's what we thought might well be happening in Leishmania. They don't have a true sexual process involving meiosis, but this parasexual process that generates a lot of aneuploidy. Okay, so I'll just describe some of the studies that are yet to be published involving a genome-wide analysis of these experimental hybrids. And we've analyzed dozens of these experimental hybrids using Illumina high-seq technology, approximate 60x coverage, And we quantitatively assess chromosome number and inheritance patterns via SNPs, single nucleotide polymorphisms. And the map reads were reference to the L major Friedland reference genome here. And the bioinformatic pathways are shown there. So for Leishmania major, this is to make the point that there is really very little heterozygosity. It's a diploid, it's a generally diploid organism, but if you look at the SNPs, most of the SNPs are by SNP single nucleotide polymorphisms between different Leishmania strains. They are homozygous SNP differences, and very few heterozygous SNP differences, okay? So very little heterozygosity. Again, suggesting that there's not a lot of sexual recombination in nature. So these are this just shows the somi of the 36 chromosomes of the two strains that we've used, the two parental strains we've used to generate experimental hybrids, and emphasizing, here's one L major strain, this is from Israel, this aneuploidy. So most of the chromosomes are diploid, but some are triploid, and again, chromosome 31 in both cases are tetraploid. Again, these parasites just tolerate this very interesting aneuploidy. So this is looking at individual SNPs across, now we're looking at just one chromosome. So I've told you, if you look here, the single nucleotide polymorphism differences between the different strains. There are over 70,000 distributed across the nuclear genome. So these bottle brush plots, we're looking at one chromosome, chromosome 17. The red SNPs indicate the SNPs that are in the Friedland, this FN parental line. These are the SNPs across the chromosome. This is the other parent. The SNPs are represented in green. Again, these are homozygous SNP differences between the two L major strains. Now, we've put them both in a fly. We've recovered a hybrid by double drug selection pressure. When we look at that chromosome, now we see biallelic inheritance. Every one of these SNP markers is now heterozygous. They've inherited at a 1-to-1 ratio, approximate 1-to-1 ratio, the SNPs from each of the parents used to generate these hybrids. So this looks like all the world to be just an F1 hybrid generated by sexual recombination, okay? And then this is a heat map, again, showing that when these things have sex, you get a fair amount of aneuploid that arises. So this color here is diploid. So these are the two parents on the top and the bottom. And again, most of the chromosomes are diploid. This chromosome 5 and this one 23 is triploid in this parent. And chromosome 31 is tetraploid in both parents. But now the hybrids, they're mainly diploid, and the numbers here show the parental contributions almost always in a 1-to-1 ratio. So now the inherited chromosomes, somes, are diploid, inheriting one allele from each parent. But you generate a lot of aneuploidy. So we think sex is probably the place where a lot of this aneuploidy arises. So that's one important consideration. So on the whole, the inheritance patterns look pretty Mendelian. Biallelic inheritance, they inherit for each of these SNP markers an allele at a 1-to-1 ratio from each parent. And that's really consistent with meiosis. So this chromosome is behaving normally. This is chromosome 19. Both parents are disomic. And this is the inheritance pattern that you would expect, and that's what we observe. This hybrid, again, is disomic, and each of these SNP markers across the whole chromosome are heterozygous for these SNPs. And then if we have a parent in which one of the parent, this is chromosome 23, one of the parent, remember, was trisomic, and the other parent is disomic, and some of the progeny, some of the offspring now are disomic, inheriting one copy from each of the parents, but some of the other progeny will have inherited 2 chromosomes from this parent and 1 from the other, and now the product is trisomic with an extra copy from this red parent. All perfectly consistent with meiosis. And this was the data that convinced us, in the absence of actually seeing meiosis and actually seeing sexual stages develop in these flies, these genomic inheritance patterns convinced us that meiosis must be occurring. But we did see hybrids that showed unexpected changes in somy, loss of heterozygosity. If we look at this hybrid, chromosome 12, both parents are disomic for this chromosome, and yet here is one of the offspring. Now it's trisomic, and it now has an extra copy, suggesting that that these aneuploid changes can arise during meiosis. And then we see total loss of heterozygosity on a chromosome in another hybrid. But these unexpected changes at a very low frequency, so just less than 1% of the hybrid chromosomes show this kind of aberrant inheritance pattern. Sometimes a partial loss in heterozygosity, probably due to a mitotic recombination event. We don't know why these unexpected patterns of inheritance occur, but again, these parasites just tolerate these kinds of aberrant somy inheritance patterns and aneuploid conditions. Then I just want to mention that the other outstanding feature characteristic of sexual recombination, of sex, is recombination. Crossover events, reciprocal crossover events involving homologous chromosomes during meiosis 1. So crossover formation is essential for the proper chromosome segregation of meiosis 1. If crossover events do not occur, normal meiotic division during meiosis 1 will not occur. So this association between homologous chromosome that leads to reciprocal crossover breakpoints is absolutely essential for these chromosomes to line up during meiosis 1. So recombination is an absolute hallmark of meiosis. So these are just describing some backcross hybrids. In the crosses I just described, because the polymorphisms were homozygous polymorphisms, SNP differences. The crossovers between homologous chromosomes that are generally homozygous for these SNPs, you're not going to be able to see these crossover breakpoints because there may have been crossovers that occurred, but because both of the homologous chromosomes are essentially have the same sequences, you won't observe them. But now we've generated a hybrid progeny that has high heterozygosity across the genome, and now we can backcross it and demonstrate whether these crossover events have occurred. So these are backcross hybrids, again taking the F1 hybrid, backcrossing it onto one of the parental strains. or taking the F1 hybrid, back-crossing it with a third strain, which has homozygous SNP differences. So these are the possible back-cross inheritance profiles, assuming meiosis and assuming a maximum of 2 recombinations for each homologous chromosome. So these are the kinds of reciprocal crossover events that can occur. And that's exactly what we found when we did the whole genome sequencing of these back-cross hybrids. Okay. And these are circus plots of these different back-cross hybrids, and these show all of the different chromosomes, the 36 different chromosomes, and the inheritance patterns and recombination breakpoints that occur within each chromosomes. So these are the parental lines. One is all red, the other green. These are the back-cross hybrids showing the inheritance patterns. And these are a physical map of the recombination breakpoints that occur in these hybrids. So this is the first physical, and we've generated genetic maps. So these are classical recombination maps that are seen in sexual organisms. And we were able to calculate the average recombination frequencies in these genomes. So there is 1 recombination event per 1.5 megabases. And these recombination frequencies are similar to what have been observed in known sexual parasites like malaria or like toxoplasma. Okay. So sex occurs in Leishmania. They clearly have the machinery for it. It is meiotic sex. Again, we haven't seen it, but this genetic analysis tells us that it really is unlikely to be anything else. And in frequencies that will be more than sufficient for classical forward genetic approaches to generate different hybrid genotypes and phenotypes to do linkage analysis for mapping studies. So we'd like to map the genes. We'd like to identify the genes that distinguish viscerotropisms and cutaneous tropisms. Still one of the most interesting questions in leishmaniasis. Why do some strains only produce cutaneous pathology and other strains disseminate to the viscera and produce these fatal infections? We don't know the answer. It's apt to be multigenic, and reverse genetic approaches, knocking out genes, putting in genes, hasn't given us the answer. It's these forward genetic, classical genetic approaches that require sexual recombination that will allow us to do it. And now that I've shown you we can cross-species hybridize a cutaneous strain and a visceral strain, we can then generate progeny, put them in mouse models, look at their tissue tropisms, do the whole genome sequencing, and generate these linkage, these QTL studies to identify the genes that associate with these different tissue tropisms. So we think this has really transformed the genetics of Leishmania now that we know that they are a sexual organism. Well, just a couple of slides to show that in the real world, that natural hybrids do exist. This is Leishmania tropica. If we did whole genome sequencing and we looked at the total number of homozygous SNPs and heterozygous SNPs, there were lines that were that showed a lot of homozygosity. These lines here, a lot of homozygous SNPs, very few heterozygous SNPs. And then there were some that had huge amounts of heterozygosity, suggesting natural outcrossing. And if we look at the distribution of these lines, it's in these Middle Eastern regions, in Israel and Jordan and Syria, that you find mainly these homozygous lines. And it's these heterozygous lines consistent with some natural outcrossing event as you go eastward, including all the way to India. That's where you find these heterozygous strains. suggesting that there was some primordial outcrossing event involving strains here, and that hybrid genotype was, for whatever reason, expanded and radiated eastward, and became the dominant strains in those regions. So if you just look at genetic markers, this is not whole genome sequencing, but multi-locus sequence typing, just specific nuclear markers on different chromosomes. These are the heterozygous lines of two strains, 747 and MA37. One's from Jordan and one's from Tunisia, I guess, or Libya. And then this strain from Afghanistan. Those markers, those allelic types seem to be, again, hybrids between these two strains. So the two parental lines had some relationship to these strains in this region. And there was a hybridization event that generated all of these other heterozygous lines in the eastern endemic zones. And when experimentally we could take the natural hybrid and do what we thought were back crosses, and we could generate these recombination maps, similar to what we did with our experimental hybrids. And again, these are recombination breakpoints, again, showing very similar frequencies of recombinations for these parasites. So for L major and for Leishmania tropica, for two different species, we've shown that the frequencies of recombinations are quite similar, and we expect this to be similar for all of the Leishmania species and strains. So these are sexual organisms. The sex occurs in the flies. Why it doesn't always occur, we have no idea, or how to optimize sex. And why it doesn't occur with insect stages in culture, we have no idea. So those are interesting questions. So the conclusions from this work: the allelic and somi inheritance patterns provide strong evidence that genetic exchange in Leishmania involves meiosis and Mendelian segregation of the nuclear genome, although the exact mechanism is unknown. The observable recombinations in the backcross progeny generated from experimental and natural hybrids indicates meiosis-like crossovers in Leishmania, and at frequencies adequate for linkage analysis and positional cloning of important genes. So important genes would be the genes controlling tissue tropism, drug resistance, virulence. So none of those genes do we understand so well. And now we can use, we hope, forward genetic approaches to get at those questions. We do see these changes in somi and loss of heterozygosity at some loci. So sex may be an important source of all this aneuploidy that we observe in Leishmania. And with that, I think we'll stop there and just take some questions, okay? So sex is fun. We wish we could see it. It'd be more fun if we could see them had sex. But it's, for genetic analysis, it's so important that we now know that these are sexual organisms.
And which are derived from the flies which are in nature.
Do they change when we culture them?
Yeah.
Yeah.
During the culture, during when we maintain the flies in the culture for a long period of time, does their virulency or in some other aspects their genetic makeup changes to an extent where they are not virulent, or later they are not virulent, and as compared to the natural strains which are existing in the natural population of the sandflies outside, which are continuously being exposed to
Right.
people.
So it's absolutely true that when we culture these things, they become less virulent. And even when we pass them, so that's well described, that culturing promasticates, they tend to lose virulence very quickly. Their ability to generate metacyclics goes down and down and down. And we don't know why that is. It has been described that that these structural changes in the genome occur. So aneuploidies do occur just during culture, okay? So we've described aneuploidy as occurring with higher frequency during genetic exchange, during sex. But it has been described just maintaining these parasites in culture, you see changes in somy, changes in chromosome copy number. And at least there's a nice paper showing that there's selection for that, that there are some increases in copy number of particular chromosome because those parasites have an advantage in growing in the culture conditions. They've increased the copy number of certain genes on certain chromosomes. And that plasticity has given them a selected advantage just to grow in culture. You know, those are very artifactual conditions. What we want to know is the nature of these changes in chromosome copy number that occur actually in the fly or in the mammalian host. And that hasn't been looked at so carefully.
And when we are meeting them in a natural host, under controlled condition, cultured condition, what do we see in the hybrid? What type of LPG we see? Because that would affect their transmission by the specific host.
Yeah, so that's a great question. So we've crossed L infantum with L major. So remember that Phlebotomus papetazi will only transmit Leishmania major. So what happened now, and if we put infantum in Phlebotomus papetazi flies, no infection, right? The infections are lost. So that is such a good question. Now we've generated a hybrid between major and infantum, and so we looked at that carefully. They sort of, some of them did survive. They had an intermediate. They didn't survive as well as the parent, but they had a sort of intermediate phenotype. But it's a nice point. In nature, if you hybridize between different species, now you may be able to get some Donovani genes expressed on parasites that can be transmitted by Phlebotomus papetazi, which we've seen even in India are in the same biotopes as the flies that transmit Donovani. So if in India Donovani somehow could have sex with L major, the genes that are essential for producing visceral infection now may be present in the Leishmania major genome and be able to be transmitted by Phlebotomus papetazi, which would be a problem, right? Yeah. So that's a great point. So it's interesting to us that this natural L tropica hybrid, we're convinced it was the product of sex between these, of an outcross between these two populations in the Middle East. And it did so well, that hybrid did so well, it became the dominant strain in Afghanistan and now even in India. So why did it do so well? Why was it so successful? Is it something about those flies that transmit them? So we don't know the answer to that. But you know, certainly the reason, sex is not obligatory, but the reason these parasites have maintained the machinery for sex is because occasionally it will produce hybrids. You know, it will mix up genes and produce offspring that have a strong advantage, survival advantage. And that's why the sexual machinery, even though it's not essential in the life cycle of the parasite, has been retained, because it happens with enough frequency and can impart such a strong selected advantage on these genomes that sexual machinery is maintained. Okay. But these parasites, you know, they have this beautiful advantage of once they generate hybrid genotypes by sex, they can then maintain themselves clonally, asexually. Sex is not obligatory. And you have to remember, sex can assemble advantageous genotypes, but if sex occurs a lot, it can also break them up. So occasionally generating it and then maintaining an asexual population that has inherited this strong genotypic advantage is probably why these parasites are both sexual and asexual. Okay, well, we'll have more discussions after lunch. Yeah. Good, thank you. So tomorrow we'll get back to a discussion of the immunology of these infections. We'll look at cell biology and innate immunity to Leishmania infection.